Hi, I'm Sheridan. Thanks for coming to my talk. Getting right into it. In response to warming oceans, the species on coastal waters are moving poleward. So to identify all the changes that are occurring on this biogeographic scale, that is entire continents, we need essentially a method that's going to have nice local resolution and be on a large scale. So recently, because eDNA has been used, especially metabarcoding, for some of these biodiversity studies, if we had a coordinated network that we could get a yearly sample from, we could track some of these range shifts over time. This, of course, assumes that eDNA can reliably recover the species ranges of the species it's detecting. So that leads us to our study group, which is the Pacific Environmental DNA Coastal Observatory, or PICO. We've collected over 450 samples over the past 2 years in the summer, and our third year is, well, starting in another week or two. We've got about 17 partners up and down the coast from California to Alaska. And one of the ways we've been able to standardize collection between the partners is by allowing them to choose 5 eelgrass sites based on their local knowledge that would be best to survey, and then giving them the instructions as to how to survey them and sending them a sample kit. So they take 4 samples 10 meters apart and a field blank. So let's, we've got some 12S MyFish data from 2021, and another set might be in my email by the end of the week. So let's look at what we've got so far. So thinking about our range shifts in latitude, one of the simple ways of thinking about our range might be the highest and lowest latitudes that the species exist at. So looking across the species that we found in the survey, we see that a certain percentage of them were only recovered or primarily recovered in fairly low latitudes. We got a nice wide range that were found pretty much throughout the entire study area. And we found another group that was primarily restricted to the northern part of our study range. And one thing you'll notice here is that in these rows, you'll see a lot more circles. Part of this is because our British Columbia samples, we just had more of them because that's where most of our network was in 2021. So we're working on improving sampling in other latitudes moving forward, but keep that in mind as we look at, as we look at comparing this data to some publicly available data through OBIS and GBIF, which have museum records from previous studies, as well as iNaturalist data. You'll see the minimum and maximum of 5 years of OBIS and GBIF data only in the summer in green, and the minimum and maximum of our single year of data in blue. So first, some bad news. We found some species that were native to the Pacific, and there was no overlap in our ranges. Similarly, we found some species that did not have any records in OBIS and GBIF in our region, and this is because they aren't there. We confirmed this with our partners, and the species that we found that didn't match aren't seen in those seagrass beds. So it's unlikely that these are invasives, and this is more likely an issue with our BLAST database or our annotation method. So we're working on addressing that. Slightly better news, we're seeing lots of wide-ranging species with ranges recovered within that BC range that we sampled a lot in. So that suggests that more sampling at lower latitudes would improve this. Next, we're seeing some that look better, and then we're seeing some that look great. So it does seem that 1 year of data is good for some species, for sure. So, and it seems like also improving annotation and all of that will help too. A quick look to the species that have the high overlap. There are 2 groups in this, one of them being a group that has many ASVs or OTUs and lots of reads, and a group that doesn't. So if you look at this more specifically, with our bar chart of ASVs and reads, just the top number, it seems that it helps to have essentially more DNA in our sample, but it's not actually required, as some of the species that have the most ASVs or the highest number of reads aren't seen in our best overlap, and some of our species that are seen in our best overlap only have 1 ASV or less than our average number of reads, which is less than 200,000. So that means there's other factors likely, well, we can discuss that afterwards, So I will wrap up because this is a lightning talk. So the latitudinal ranges are looking good compared to our publicly available data, and you can't just have a lot of reads and a lot of ASVs to match your ranges. So thanks for listening.